How's it going, everyone? It may not be obvious to some, but the reality is we have a major problem on our hands. Technology has become deeply integrated into our lives, and trouble has been developing right in front of us. With the proliferation of personal computers, smartphones, and data-heavy processes, the amount of data needing to be stored has started to stack up, and we will quickly outpace our current data storage capacities. For a point of reference, the Library of Congress is one of the largest libraries in the world. If you were to scan and digitize all the books and documents in its catalog, the total amount of storage needed would be anywhere from 10 to 20 petabytes, or 10 to 20 million gigabytes. To give a visual on the sheer amount of data in the Library of Congress, if you were to print all the data on standard printer paper, it would amount to 5 to 10 billion pages. If stacked, it would be 311 to 200 or 622 miles tall. For comparison, the orbit of the International Space Station is only 250 miles high off the surface of the Earth. So what are we doing now to store all this data? Currently, we're brute forcing it by simply using more and more hard drives. Over the past few decades, data centers have been built in order to store large amounts of data in one secure location. They have their cons, though. They're expensive to build and maintain, consume huge amounts of energy, and require large amounts of land to build on. Hard drives are also prone to failure. Nature also faced a data storage problem. Its solution, DNA, using a brilliant system of four nucleotides abbreviated simply as A, T, G, and C. Despite being millions of years old, it still proves to be a useful method for storing impressive amounts of data. But what if DNA could be manipulated for our own digital needs? Luckily, it can be. Researchers have been developing methods in order to convert our digital data into strands of DNA. The general process begins by taking data, converting it to binary code, converting that binary code to nucleotides with the use of an encryption key, and synthesizing a strand of DNA with the data encoded in it. The sequence is then embedded into a bacterial genome. Here, the bacteria with the encoded data is stored for later retrieval. This translation process is streamlined by developing software known as a DNA codec. Two groups of scientists have shown promising success in this field, developing and utilizing novel variations of the methods that were just described. Yache et al., using Bacillus subtilis, encoded the message E equals mc squared in 1905. This is also a member of the human gut microbiome, which presents some interesting potential applications in the future. A separate group of researchers, Yim et al., used E. coli BL12 to encode the message Hello World. So out of all the materials, why DNA? There are a few specific reasons. DNA is a universal material. Because of its fundamental place in scientific research, the needed technology is here to stay. There are also a variety of species of bacteria that can be used. DNA, when stored properly, is also stable. Another reason is security through obscurity. It's hard to steal data if you don't even know it's there. The main reason, though, for using DNA is its data density. It's estimated that 1 gram of single-stranded DNA can hold 455 exabytes of data, or 455 billion gigabytes, enough to hold the entire contents of the Library of Congress 22,750 times over, all able to be stored in a test tube. There are some drawbacks, the first being how expensive it is. It requires specialized equipment that costs hundreds of thousands of dollars to read, write, and store the encoded data. The data recording speeds are also very slow. There's also an inherent risk of data corruption due to mutations as well as horizontal gene transfer. So what are some possible uses for this technology? The most obvious is to build a purpose-built data center that stores large quantities of bacteria with embedded data. This is a huge improvement on current data centers as they could hold the same amount of data in less space. This would be perfect for archival data storage, data that you want to keep but don't need quick access to. Leaning more into the sci-fi side of things and looking further into the future, the use of species found in the human microbiome, such as Bacillus subtilis used by Yache et al., presents an incredibly unique use case for a true personal data storage solution, able to store large quantities of data in or on your body. Though this is far in the future, but interesting regardless. This field is still incredibly young and very much in its infancy. Methods are still being developed and tested. Standards for this field are still being determined to create a unified path forward to bring this technology into practical application. But with multiple successes, it's showing a lot of potential as a solution for society's growing data storage problem. Here are my references, and thanks for watching.